Good afternoon, everybody. On behalf of Milan Merksic, the Vice President for Research, and Eric Nielsen, the Vice President for Health Affairs, and Louis Landsberg, Dean of the Feinberg School of Medicine, I welcome you all to the CLP-Feinberg Convergence Workshop. As I suspect most of you know, the Institute was created in 2005 as the outcome of a 2005 to 2010 strategic planning process to stimulate interdisciplinary biomedical research across all of Northwestern colleges, schools, and hospitals, with a particular focus on the fundamentals of chemistry underlying biology. Initially, this was the idea of then Provost Larry Dumas, and the Institute began programming in 2009 when its new home, the Richard and Barbara Silverman Hall of Molecular Therapeutics and Diagnostics, opened its doors. Since then, the Institute has grown from a founding group of 6 faculty members, 1 staff person, and limited research support to more than 60 tenure-track faculty, 14 research faculty, 15 administrative staff, and 55 technical staff and research associates. Currently, its major focus of research areas are understanding the human proteome and how to deploy that platform to fight human disease, developing non-genetic strategies to control cancer, assessing and treating diseases through theragnostics, decoding the role of metals in human health and disease, and accelerating team science. One of the key goals of taking the Chemistry of Life Processes Institute to the next level is deeper engagement with the Feinberg School of Medicine and its scientists and clinicians. And that's really the goal of today's event. So I thank all of you for your participation and active engagement. It should be an interesting 3 hours, and we hope it'll stimulate many, many further conversations. So without any further ado, I'm going to introduce you to Neil Kelleher. He probably needs no introduction to most of you, but let me mention a few of his important roles. He is the Walter and Mary E. Glass Professor of Molecular Biosciences. He's a professor of chemistry. He's a professor of medicine in the Division of Hematology and Oncology. He's director of Northwestern Proteomics. And most important for today, he is the director of the Chemistry of Life Processes Institute. Neil, floor is yours.
Thank you so much, Rex, and welcome everybody to the Convergence Workshop. Well, we'll workshop some ideas and some things that you may be aware of and can get updated on, or some new things that might lead to new outcomes. And one of those is more team science. And so let me go through a few matters of bookkeeping and business, and then we'll jump right in. We'll have 2 sessions, and I'm really thankful to Stephen Deitcher and the Family Foundation for helping us get this together. And in these 2 sessions, we're first going to hear from Feinberg faculty and see what's new, what's on their minds, what their research programs are doing, and what they aspire to do. And in the second session, we'll focus on technologies and platforms that are differentiated to help compete on focused biomedical problems of our day. Each speaker will give a brief overview. They're designed to be brief, and that'll help catalyze the Q&A, which will be a lot of fun. And each speaker panel as a group will address questions after each session. So we'll roll through the speakers and then have Q&A. We'll have a 20-minute break in between, and then reconvene, followed by a dinner this evening. And there are a few slots left, so we've got about 25 for dinner tonight lined up. And audience members can enter questions in the chat anytime during the program. The first session will be moderated, the Q&A there, by Sue Quaggan, and the second by Carl Scheidt, who can field and stimulate questions in Q&A after each of the 2 sessions. And then everybody will be unmuted in those Q&A sessions. So it'll be free form, and you're welcome to chime in via chat or directly. So yeah, our goals have been formulated to really reconnect with the unmet clinical needs of our day, disease models, translation. This is always of interest to those developing methods, the physical scientists, many of whom are in Evanston. So bridging the 13 miles and also bridging genotype to phenotype is another theme as well. And so we want to share unique and differentiated Northwestern technologies and find places where we can compete and foster new transdisciplinary collaborations, team science to advance new lines of grant applications to federal sources and foundations, but of course always driven to try and improve patient lives and research environment at Northwestern. So to follow on briefly, here's the current landscape of CLP and your favorite pie chart there on the left. So it does touch across the university, many schools and colleges. So we have a lot of advisors, and I really want to thank the Feinberg SAC, or the Scientific Advisory Committee, that was formed when I became director about a year ago. And they've really helped steer in a series of meetings that was held a while back. And there's been action out of that and competitive grant submissions. So we want to expand upon that. And we also thank the Executive Advisory Board, of which Stephen Deitcher is a member, as well as the Faculty Advisory Committee, who has a meeting next month for the Institute. So yeah, what do we do in CLP? Molecular therapeutics and diagnostics. It is written, as Rex will remind me, on the side of the building. So that's certainly what we do. But we're trying to update that and try to focus parts of the Institute to be more poignant for the translational mission. And that means clinical research as well as basic research, and connecting both more tightly across the 13 miles that separate us. So differentiated technologies, that's an important aspect of it, and not just to establish the technologies, but to really reduce them to practice into truly next generation platforms to accelerate discovery, and not just differentiate the technology, but differentiate Northwestern. And we have a vibrant training cohort of future leaders. We have a NIH training grant. And I thank Amy Rosenzweig and Regan Thompson for helping me, along with Rick Silverman, who started that program some years ago, train all of the next generation. So there's a lot of discovery opportunity through robust platforms, chemical and top-down proteomics, a variety of imaging, a subset of which you'll be hearing about in the second session. Molecular modeling and computation. That's something that in the February 8th Feinberg retreat was pointed at as an important part of Feinberg's strategy over the next 5 years, bring in computation. Of course, small molecule inhibitors, expertly designed. And so you'll hear from Carl and about the rich tradition of that at the Institute and at Northwestern. We have a fountain of new natural products coming from fungi and bacteria, and all while doing HTA and high throughput screening, as Al knows very well. And so we've got a lot of areas of collaboration. We've submitted a fair number of grants across disease areas in oncology, cardiovascular, immunology, immunobiology. I'm really excited to hear from Satish later. Of course, neurodegeneration and the brain initiatives at CLP has been running for a year or more with Hande and Rick and myself, and that's gotten some DoD grants. And aging and others will be determined. And so that aging makes me think of Doug, and this will be my last slide. So I'll hand it over to Doug to take us into the first session and the goals there on that slide. So please, Doug, take it away.
Thanks very much, Neil. I hope thinking you don't think about me in the context of aging because I'm old.
Wait, that came out wrong.
Yeah, it came out very wrong. Let's try that again.
I've got more gray hair than you, Doug.
Listen, I'm really happy to be here and excited about the opportunity to talk about potential new areas of collaboration and discovery on campus. I'm here representing the Department of Medicine and as the chair of that department, and also as the inaugural director of the Podesnak Longevity Institute at Northwestern. And I'd like to say a few words about both of those entities that kind of give us some context about where we are and where we might find new avenues of collaboration. The Department of Medicine is a large clinical department, but we have a substantial research portfolio that spans from very, very basic science at the molecular and cellular level, all the way to population science, outcomes research, and even implementation science. Our portfolio is rather diverse in terms of content as well. We have large research programs that are supported by the Heart, Lung, and Blood Institute, the National Cancer Institute, the National Institute of Aging, NIDDK, and others. The opportunities for collaboration, and particularly in the context of CLP that come from the Department of Medicine, I think are largely reflected in the fact that many of our investigators and much of our research is targeted towards the discovery of pathways that contribute to disease and discovery of new targets that might be amenable or might represent therapeutic targets that can provide some new ways to treat disease. So there are already a number of interactions between the Department of Medicine and CLP that Neil mentioned a moment ago, and we would like to see those expand, grow, and synergize in the next several years. The Podesnak Longevity Institute was created early this year by John Podesnak and his family with a generous gift to Northwestern. The Longevity Institute is built around existing programs in the field of aging. We have a substantial research portfolio from the National Institute of Aging here at Northwestern. As of last year, we were over $40 million total in research funding and ranked in the top 15 in the country in terms of total funding from the NIA. What the Longevity Institute wants to do, though, is to try to coalesce and coordinate a diverse set of investigators in a variety of different areas that speak to aging and aging-related morbidities. We have created 5 new centers as part of this institute. The first is a Center for HIV and Aging, and that's one of the cornerstone institutes of or centers of the program. We also have a Center for Basic and Translational Science and Aging. We have a theoretical or aspirational center for nanoscience and aging that I think touches on a real strength of Northwestern. We have a Center for Population Science and Aging. And all these centers are really designed to help coalesce and coordinate existing activities among a variety of investigators, both downtown and in Evanston, that address issues related to aging and longevity. We hope that by creating these centers, we'll have opportunities for new collaborative research and for synergy across a variety of different domains. There's one entirely new center that's being created. It's called the Human Longevity Laboratory. That's a place where we'll bring in adults from the Chicago area or outside of Chicago and precisely measure their biological age in a variety of different dimensions. This unique resource, I think, is likely to provide us with substantial opportunities to think about how to develop new drugs, how to measure aging and quantify it precisely, and also give us a chance to start to think about interventions that can either slow the pace of aging or even reverse aging in adult humans. So it's a very exciting time for us. We hope that the Podesnak Longevity Institute will give us an opportunity to take a leadership role role in the field in that domain in the years to come. And we expect to have a number of different opportunities to interact with people through the PLI and the CLP. I think we'll go ahead now with the first session. And I'm happy to introduce our first speaker. Satish Nadig is our Chief of Organ Transplantation in the Department of Surgery. He joined Northwestern almost exactly a year ago and has already reinvigorated that program in remarkable ways. And I've found him to be a fantastic partner and collaborator, both in the clinical realm and in research as well. So I'm happy to introduce Satish to everyone that hasn't met him yet. He's a great addition to our campus. Satish.
Thanks so much, Doug. It's an honor to share the screen with all of the folks on this call and all of the collaborative minds that are represented here. Next slide. Am I to share?
Yes, good. If you've got your slides there, Satish, you
Sure.
share it. Will.
Okay, give me a sec, because I had submitted them, but I'm happy to share them here. So.
No worries.
So, I have 10 minutes to tell you why we are way behind the 8 ball in transplantation, and to tell you how we're on the cusp of the next era of transplantation, if you will. So, as you all know, cardiac allograft vasculopathy, transplant vasculopathy, chronic rejection, these are all immune-mediated phenomena that are modified by non-immunologic factors and cause this fibroproliferative lesions that occur in the organs, in the vessels supplying these organs. So if you can imagine an organ on the end of these vessels that are riddled with these vasculopathic lesions, they end up undergoing fibrosis and dying. And we just don't have a cure for this. It's the rate of chronic rejection in organ transplantation is the same as it was in the 1960s. And when I was doing my PhD in the mid-2000s, we realized that we could actually expand these regulatory-based T cells, which we all know now, but at the time they had not been used in humans. And we created this model where we took these immunodeficient mice and transplanted them with human arteries and gave them human immune systems, and were able to replicate this vasculopathy, or this pathologic hallmark of chronic rejection, and then expanded these regulatory T cells ex vivo and put them back into these mice as therapeutics, and minimized and or abrogated completely this vasculopathy. What's interesting is that regulatory type cells obviously have expanded in their scope, and now at Northwestern we have more people off anti-rejection medication than anywhere else in the world. 37 patients off anti-rejection medication, because we're taking some of these same things that I did in the lab many years ago, and Dr. Leventhal and team are expanding these regulatory T cells ex vivo and putting them back into patients, and it's working. But we still have to give systemic immunosuppression. And that's the problem. And so one of the things that we're trying to pursue is, how do we precondition these organs with anti-rejection medications, some of which are very good, like rapamycin, into these nanoparticles, and target them to the organ so that the patient doesn't need anti-rejection medication systemically, but the organ is immunosuppressed. And so we can take these drugs that are encapsulated in these stealth packages and deliver them directly to the area of need, and their control released. And we want to know, could we focus this mTOR inhibition, which is the best type of anti-rejection medication, but seldomly used perioperatively, because of all the side effects, could we harness their ability to expand these regulatory T cells by putting them into a micelle nanoparticle and delivering them to an organ? We call this a targeted rapamycin micelle, or TRAM therapy. Well, interestingly, I just put in 2 kidney transplants today, and they came from various parts of the country, and they come out of a donor patient. They go into a cold solutions that were developed in the 1980s, by the way. They go into a plane. They get transported here in preservation, and then we take them out, and then we start patients on anti-rejection medication after we've sewn these organs in. And people are still just debating on what organ preservation solutions are the best solutions to use. Well, here's the thing. We're using anti-rejection medication in the current era that were developed in the 1970s, 80s, early 90s. We're using preservation solutions that were developed in the 1980s. And we just haven't moved the needle, and we're suppressing a person's entire immune response in order to suppress the organ when we have this whole time period where an organ is out of the body for a period of time where we can target specific cell types like endothelial cells, for example, which is what we're particularly interested in as the first line of defense in these organs, to suppress, to mitigate the systemic immunosuppression used. So our solution is to take these TRAM enhanced preservation solutions and pre-treat organs to downregulate these rejection responses. So what we found is we could, by targeting these rapamycin micelles, we could target these endothelial cells in a very specific way, as opposed to untargeted RAM that you see on the left. When we put it up against free rapamycin, which by the way is used and FDA approved, and we could potentially have a point of collaboration here in the Longevity Institute with rapamycin. But when we put a toxic MPER on these cells, you kill the cells, but when then you compare their toxicity to free drug across escalating doses, they have the same toxicity profile as free drug. And they immunosuppress. We drop down levels of harmful cytokines like IL-6 and IL-8 when that are secreted by the endothelial cells when injured. But when treated with this nanoparticle, it's decreased. And when we give it to these aortas and these mice at escalating doses, you get dose-dependent accumulation in the tissues, as you can see from the top line, compared to just our University of Wisconsin solution alone, or just preservation solution that's the standard of care and has no therapy. So what if we took these arteries from certain mice, incubated them in a solution that's enhanced with our rapamycin micelles, and then transplanted to genetically different mice after storage for 6 hours, which is about the average time we store organs, and then analyze them for this hallmark pathology of chronic rejection. Well, first, if we do twin mice isografts, we get no rejections, our negative control, and positive control allogeneic with the standard of care University of Wisconsin solution alone. You just take free drug and dump it into the University of Wisconsin solution at high doses, 1,000 nanograms per mil, really doesn't do much to the vasculopathy. But at 1/10 of the dose targeted to the endothelial cells and encapsulated into a nanoparticle, you get almost complete abrogation, if not complete abrogation, amongst multiple animals and repetitions. And so you see that the targeted rapamycin micelles are very similar to the syngeneic grafts and significantly better than free drug or, of course, the empty micelles. But then a drug is not a drug unless you decrease the dose of it to show that it is dose dependent. And so when we drop that tenfold, we lose that tolerance. And so that was really exciting for us. And we've now patented this and started to go into large animal models. And so a real frontier in the next generation of immunobiology is targeting these drugs to the organs rather than the patient to get them off the anti-rejection medication. And how is that linked to the regulatory T cells? Well, we know that this rapamycin expands regulatory T cells, and we know regulatory T cells work because we're already doing that in clinical trials. But what if we were to expand it in vivo in the organ itself to create a microenvironment of tolerance, if you will? Along the way, we have to figure out what the mechanism of this was. And what was really interesting, and this is a picture of Dan Tran, my MD PhD student, who's now a medicine resident at the University of Virginia. We asked, what are we doing here? We're taking a warm organ, putting it into a cold solution, traveling across the country, and then putting it back into a warm body. But nobody's looking at what happens to the immunometabolism of these organs and specific cell types when they're in these cold preservation solutions upon ischemia reperfusion. So we asked, does immunometabolism play a role in the mechanism of protection of these nanoparticles. And nobody had really seen this in the endothelial cells, but they had done work in what do dendritic cells look like from an immunometabolic perspective when injured. And interestingly, they adopt glycolysis, and they have a decreased reliance on mitochondrial metabolic machinery. And the bottom two figures, this is what endothelial cells are doing when they're injured. In cold ischemia, in the first 6 hours, they're highly dependent on glycolysis and decreased dependency on mitochondrial metabolic machinery, which is seen on seahorse technology, and which is not surprising, very similar to professional antigen-presenting cells, as they are semi-professional antigen-presenting cells. So when we recapitulate what happens in the clinic, in vitro, and take mouse endothelial cells and we incubate them with in a cold hypoxic chamber and reperfuse them, and use that time to treat them with drugs like rapamycin, interestingly, what we found was you have normal cells that are injured have rounded mitochondria, which you see in the red punctate mitochondria, which we all know from the textbooks. But when treated with rapamycin, these cold ischemic cells, the mitochondria fuse, and they form this tubular shape. So does mitochondrial morphology correlate to this immunogenicity? And can we force mitochondrial elongation if that's good? And so we looked at the cancer literature, and Erica Pierce and colleagues have done much work on what happens to T cells when you force their mitochondria to fuse. And they do alter their behavior. And there's particular genes, for example, DRP1, that causes them to undergo division, and mitofusin 1 and 2 and OPA1 that causes them to fuse. So mitochondria are dynamic, as not surprisingly. They're, depending on the environment, they're fusing and dividing rapidly. So we decided to use a pharmacologic, M1, MDivi1, a fusion promoter and fission inhibitor, and force fuse these mitochondria. And when we took mice and injected them with an allogeneic endothelial cell strain, and then sorted out CD8 positive memory-like T cells from these spleens, and then co-cultured them into endothelial cells that were pre-treated with just mitochondrial fusion pharmacologics, we looked at what the T cells phenotype of injury was after treatment. And when just cold stored and warm reperfused endothelial cells that are co-cultured with T cells produce massive amounts of granzyme B and interferon gamma. But this is completely abrogated when we fuse the mitochondria of the endothelial cells, suggesting that their antigen presentation is quiesced upon reperfusion. So when we did this, when we treated these endothelial cells before they go into a cold hypoxic chamber and then co-cultured them, as we showed you before, but then blocked PD-L1, we found that this was PD-L1 dependent, because we were able to reverse that effect upon PD-L1 blockade. So interestingly, we wanted to see if this translated to in vivo, and we took hearts out of mice, transplanted them into allogeneic mice, and in these controls, you get massive amounts of lymphocytic infiltrate. But when we gave the donors M1, MDivi1 and fused their mitochondria, we got normal hearts 24 hours later, which was pretty interesting. When we actually put that into a more robust animal model, where we have a model that is clinically relevant, a brain death model, we induce brain death in a mouse, confirm brain death, then give mitochondrial fusion pharmacologics, pre-treat the donor for a few hours, heterotopically transplant the mouse with the heart, which is what a standard of care, a standard model for immune rejection is, and then look to see if we can see what happens to survival. We took the histology of the mouse, and we found that brain-dead control mice have massive amounts of VCAM production, but when treated with mitochondrial fusion drugs in the donor, we're able to see really not much VCAM at all. So showing that the co-stimulation is also affected in antigen presentation in vivo. So when we did these brain-dead control mice, we got early rejection. We also treated them with antioxidants, so N-acetylcysteine, to show that it's not what happens when you just inhibit the reactive oxygen species, and not much in this model. But we're able to significantly prolong survival just by fusing the mitochondria. Not long-term, but significantly prolong it such that what we're doing now is giving subtherapeutic doses of immunosuppression to see if we can induce long-term survival. So really the idea is we need to do better in altering the metabolism of these organs. And so people are working on it with normothermic perfusion, but the standard of care right now is cold storage. And how do we precondition these organs prior to going into patients? So we think that focused nanotherapy could be successfully added to these preservation solutions and abrogate vasculopathy, and then potentially altering mitochondrial dynamics or various immunometabolic pathways could be used as a druggable target for conditioning of these organs, with the general theme of pre-treatment rather than treating systemically the patient at the time of transplant when we think we're behind the 8 ball. So these opportunities could pave the way for future trials in donor organ for treatment. And so we're partnering with Evan Scott, Sam Stoop. We've gotten a few grants to translate this into a very unique model where we take discarded human organs, put them on a pump, and use that as preclinical data. This is our lab. We did a lot of this work in my previous institution in Charleston, South Carolina, and in purple are the people that have come here with me and really do all the work and some of our funding mechanisms. And this is just a photo of our lab and just a recent book that one of our previous colleagues here at Northwestern and I published, Jason Wertheim and I published, talking about all the different technological advances in organ transplantation. You know, what we are really on the cusp of the next era of transplantation, and I call that the era of technology. And I think Northwestern's on the forefront. So I appreciate the 10 minutes to tell you why we're behind the 8 ball, but why we're in the leading edge.
Thank you very much, Satish. That was tremendously exciting, and we look forward to great things from your group and the transplant program. We'll save questions for the a question and answer period at the end of the discussions today. So thanks again, Satish. Next, I'd like to introduce our next speaker in this afternoon's program. Dr. Al George is a professor of pharmacology and medicine and chair of the Department of Pharmacology. And Dr. George is going to talk to us about some interesting research topics in the field of cardiovascular disease. Al?
Thank you, Doug. Thank you very much. I appreciate the opportunity to share some thoughts about cardiovascular disease research at Northwestern. I want to thank Neil Kelleher for his leadership in CLP and also putting this convergent workshop together. Hopefully it's the first of many. So my approach to this was to not highlight specifically my research, but to give a broader context of research that's ongoing throughout the Feinberg School of Medicine. So let me begin by just saying this is about cardiovascular research, which by definition means things related to the heart and blood vessels. And these are some of the many topics of active research ongoing among faculty at Feinberg. And I'll highlight a couple of these on the heart side. There are many people who are much better at giving voice to the elegant and really leading work done in the area of vascular biology. There are many centers at Northwestern that focus on cardiovascular research and cardiovascular disease. I hope I've caught and listed most of them. And as a testament to the reputation of cardiovascular research at Northwestern, I put up this slide, which is a map of universities and other research centers around the US that have received funding from the American Heart Association in one of their new programs called Strategically Focused Research Networks. These are multi-investigator collaborative grant programs that have been given out over the last decade or so in different themes. And down below here, at the bottom of the map, you can see the various themes that have highlighted each year of the program. There's been a theme on, for example, children's health, vascular disease, atrial fibrillation, arrhythmias and sudden cardiac death, and others. And I just want to highlight the fact that Northwestern has received more of these SFRN grants than any other place in the country. And I think that just is, again, a growing testament to the the reputation that we have in the country as to the quality and depth of our research programs. I'm part of the Arrhythmia and Sudden Cardiac Death program, along with Beth McNally and other investigators at Northwestern. And I wanted to highlight some of the work that we've been doing as part of what we call the Northwestern Sudden Death Collaborative. And this is a union of several investigators, all of whom are listed in this authorship list on this paper, that has been funded for the last several years by two non-renewable grants. The first was an NIH cooperative agreement, or UO1 grant, that was designed to investigate sudden death genomics in a population or a registry of sudden death in the young. And then separately from that, Gregory Webster, a cardiac electrophysiologist at Lurie Children's Hospital, who's currently funded by a K-23 grant from the NIH, independently assembled a registry of a couple of hundred older individuals that had sudden death, still under the age of 40, but not infants as were in the SDY database. And our focus has been to use the power of whole genome sequencing to delineate genetic and genomic factors that were associated with sudden death. And with the leadership of Beth McNally and the Center for Genetic Medicine, we now have more than 1,000 whole genome sequences, both on healthy individuals and folks with various disorders, including more than 300 cases of sudden unexplained death, and at least 100, if not many more, of individuals who have a familial cardiomyopathy. And one of the long-term goals of our AHA program is to develop a polygenic risk score that can be a valid way of predicting who is at greatest risk for sudden cardiac death. Another area of interest of ours and other several investigators at Northwestern is to use human-induced pluripotent stem cell models of cardiovascular disease. And this is linked in very much with our AHA program to actually model sudden death risk in cardiomyocytes derived from iPS cells. For example, as many of you know, iPS cells can be grown in two-dimensional culture, and they can beat, just as I'm showing in this image. And we can take cells and create iPS cells from healthy individuals and those with a particular disease, whether it's cardiac arrhythmias, cardiomyopathy, or some other complex phenotype. And then we can not just make cardiomyocytes, but through the leadership of Paul Burridge's lab in the Department of Pharmacology, he's also blazed a trail in showing that one can make high quality endothelial cells to model vascular disease, as well as vascular smooth muscle cells and fibroblasts. And then subject these cells to a variety of high throughput technologies to measure the contractility, for example, of cardiac cells or vascular smooth muscle cells, to perform electrophysiological experiments in high throughput, to look at intracellular calcium dynamics, and to use these platforms for drug screening. Beyond two-dimensional cultures, there's now evolving technology to develop three-dimensional engineered heart tissues. And this is work that's being done in Beth McNally's lab, as well as a new faculty member, Igor Efimov, who I'll say a little more about in a moment. These technologies have been exploited by many investigators here. I just want to highlight the work, some recent work published by Paul Burridge's lab, where they studied the genetics and molecular basis for a chemotherapy-induced cardiotoxicity that is well known. And they identified two genetic determinants and then used the iPS cell technology to explore the underlying molecular and cellular mechanisms. And from both of these studies, they identified new targets that could potentially be drugged in a way to prevent this toxicity from occurring. And one of those candidate drugs is a repurposed FDA-approved drug that they're beginning the process of developing a clinical trial here at Northwestern. My lab has had a longstanding interest in using electrophysiological techniques to interrogate all kinds of cells, including cardiomyocytes. And when I came to Northwestern in 2014, we installed the first 384 well planar patch clamp system in the US. We've recently had the opportunity to upgrade this system and have used this to automate the electrophysiological interrogation of iPS cell-derived cardiomyocytes. And this is work done collaboratively with Paul Burridge's lab, with the great assistance with Carlos Benoye, a research professor in our department. And this is just an example of the readout from this technology. These are patch clamp experiments done in 384 well plates with high fidelity and robust data quality. And here we can easily measure action potentials from cardiomyocytes. I won't say easy, but now it's easy. It took many months to optimize the technique. But more than that, we can measure action potentials, sodium currents, and calcium currents in sequence from the same cells in an experiment that lasts less than half an hour. And so this is a great technology that I think we've recently had some breakthroughs with, and I think this is going to pay great dividends going forward. We've already used this platform to measure sodium current to evaluate a candidate gene for a disease called Brugada syndrome that was recently identified by an international collaboration that used a genome-wide association analysis to find new risk loci for this gene. One of the genes, MAPRI2, which is the N-binding protein 2, and part of the microtubule network in cells, turned out to be a risk factor. And by knocking it out in iPS cells, we could demonstrate that cardiomyocytes have impaired sodium channel function. A new member of our faculty on the medical campus, but a member of the biomedical engineering department, is Igor Efimov, who is a well-known and highly regarded investigator in the biology of arrhythmias. He's made his mark many ways, one of which is to use techniques of optical mapping of cardiac arrhythmias in both rodent heart and human heart. And he's now exploring new technologies to do high throughput interrogation of engineered heart tissues. He also collaborates deeply with John Rogers, who the team of Igor and John and others at the medical school have explored the development of implantable and resorbable cardiac pacemakers that will potentially transform temporary pacing following cardiac surgery and other applications. One of the investigators on there was Rishi Arora, who independently has a research program designed to explore new therapeutic strategies for the most common cardiac arrhythmia, which is atrial fibrillation, which is a high risk factor for stroke and other cardiovascular morbidities. He's developed some novel gene therapy approaches targeting the NOX2 gene to downregulate certain ion currents in the heart to normalize atrial electrophysiology in a way to reverse the risk for atrial fibrillation. And while on the topic of atrial fibrillation, a new research center for arrhythmias at Northwestern, led by Rod Passman, was just awarded a very large NIH clinical trial grant to evaluate the use of wearable technology, in this case, the Apple Watch, which can be used by individuals to detect atrial fibrillation as a way of testing whether or not this technology can be used to trigger a patient-directed anticoagulation therapy to prevent stroke. Again, wearable technology interacting with healthcare system is a new and exciting technology. So that was just really a quick and dirty overview of just a slice of the deep cardiovascular research areas that are active at the medical school. So thank you.
Thanks very much, Al. Tremendously exciting and diverse cardiovascular research here. Our next speaker is Daniela Ladner, Vice Chair for Research and Innovation in the Department of Surgery. And she's going to talk about challenges in liver disease and transplantation research. Daniela.
Thank you very much. I'm going to share my slides here. Just want to confirm that you can see those slides.
Yes.
All right, I thank you very much for letting me talk to you a little bit about liver disease, specifically cirrhosis, and as it pertains to liver transplantation. I was going to talk to you a little bit about prevalence and the risk and mortality and predictors, and then our opportunities I see in this area. You know, the liver is near and dear to me and has many properties, including the ability to regenerate, something that the ancient Greeks seem to have known. If you know the story of Prometheus, who was shackled to a Caucasus rock, and an eagle came to eat his liver every day, and it regrew at night. And the truth is they didn't really know that the liver could regenerate, but they kind of by accident got it right. But amongst the many properties of that the liver has is not just regeneration, but it's important for our protein production. It's important for glucose storage. It's important for lipid metabolism. It's something really central without which we cannot live. Therefore, if the liver is tremendously diseased, we need to replace it through transplantation. In terms of cirrhosis, which is basically the disease of liver when the liver gets replaced by scar, the prevalence of cirrhosis has tremendously increased just over the last decade. To a large part, that is due to the growing problem of obesity combined with hypertension and diabetes, but also alcohol use disorder, hepatitis C are large contributors, as well as biliary cirrhosis and so forth. But it affects probably about 2 to 10 million adults in the United States. And you might be like, well, that seems a very broad range. The truth is we don't exactly know. If we look at databases, it's closer to 1%, but in random autopsies of diseased patients, we find it up to 4.5%. Either way, it's not a small portion of the population that has this disease. When you are diagnosed, once the liver is scarred enough to qualify to be called cirrhosis, which means most of the functional tissue is replaced by scars, your median survival drops down to 12 years. But what's worse even, if you then have a decompensating event, such as bleeding, such as confusion, which is called hepatic encephalopathy, or water in your belly, which is ascites, once you have such decompensated complications, your median survival drops to less than 2 years. And you can see this on your survival curve here to the right. And the reason is really once it's decompensated, it becomes a systemic disease with multi and system organ dysfunction. What's really important, and I think here are the opportunities for any kind of investigation. Patients, they transition from compensated to decompensated at a rate of about 5 to 7% every year. That's a pretty significant difference. Why should you care? Once a patient is decompensated, their need of hospitalizations, their need of healthcare resources increase tremendously. These are not patients who just then die. They require tremendous resources, but ultimately, if they do not get a transplant, they will die from their liver-related disease. The other thing that happens at a fairly significant rate is hepatocellular carcinoma. Just like when you have burn injuries and scarring of the skin, when you have scarring of the liver or injury to any other organ, you are susceptible to cancer. And patients with cirrhosis, they have an incidence of cancer every year of 1 to 8%. So on average, you could say about 4% of patients who have cirrhosis every year are diagnosed with a new cancer. And I just told you there are about 2 to 10 million people in the country who have cirrhosis. That's a significant amount of patients who have cancer, many of which are not diagnosed until they're very progressed. So to go back to kind of the categories I mentioned before, you have compensated cirrhosis, so patients who have scarring of the liver, but kind of go about their daily business. Usually there's some fatigue, but once they decompensate, they're usually really significantly impaired in their function, in their ability to hold down a job and require additional resources. One of the really big questions is, so among all these patients, who are the ones that are going to decompensate or die early? Who are the ones who going to develop cancer? So if we look at mortality, it's been the attempt to figure out who are the patients at greatest risk over the last 70 years. Child Turco, for those of you who are clinicians, there's the Child Pew score that tells you what your risk is in terms of dying from your underlying disease. That was built based on some lab values, but also on a clinical assessment to see if patients are confused, if their nutrition is adequate, if they have water in their abdomen. That has advanced to a score that allows a 3-month mortality. You might be like, well, why does that matter? Well, in the United States, the allocation of the liver is based on the sickest first, and a not so gameable way of identifying who's the sickest is making it dependent purely on labs, so not on a clinical assessment. Why is that? Because as clinicians, we will probably always push for patients to be most likely to get a transplant. And so to get the gaming out of this, a score was created based on clotting and the kidney function and the production of bile in the liver. It's called the MEL score. That MEL score got amended, so to speak, by also including sodium. Turns out if the liver doesn't work really well, your sodium in the body drops, and that's also a predictor of mortality. But the issue is that most patients with cirrhosis actually have a low MEL score. Just for those of you who are not familiar with the MEL score, it starts at 6, that's the lowest score, and the highest score is 40. And as I said, the score is associated with a mortality rate. The rate of 6 has a mortality rate of way less than 1%. It's kind of the mortality of that age group in the population. But a MEL of 40 has a 3-month mortality score that is close to 80%. If you look at these graphs on the right side, though, you can see that if you're in the lower MEL score, let's say below 20, most of the patients, not all of them, are compensated, but there are still a significant amount of patients who are decompensated. And I mentioned earlier, it's the decompensated patients that are likely to require hospitalizations, additional care, and are likely to die. And so the MEL itself, while a fantastic predictor of 3-month mortality when you're really sick and your MEL is high, is not really a very good predictor of mortality when the MEL is low. And so again, what you'll see here on the lower quadrant, you see the MEL distribution by age. This is really a disease of the elderly. But again, you will see it's those patients with low MELD scores that are much more frequent. Now, what has been clearly shown is when your MELD is low, it is no longer a good predictor of mortality. In fact, it kind of becomes close to a coin toss. So the MELD score that's used in transplant doesn't really predict nicely who the people at risk are in the greater scheme of things, greater cohort that have cirrhosis. Yes. In terms of the mortality rate, you might be like, well, okay, so these patients with lower MELD scores, you're saying, well, they're not as sick and they might be dying from other causes. We actually looked at this and we found that while obviously the mortality rate is much lower in patients with a lower MELD score, still the predominant reason for dying, and even in the lower MELD score at 47% is due to liver-related causes, whereas obviously when the MELD is higher, it's 70% that it's liver-related. So there's a lot of investigation going on here to try and figure out what the likelihood is of these different states. There's been a study that really has been what we've been working on for the last 30 years out of a population from Italy. And that graphed on the left side are the percentages and the likelihood of going from one stage to another. But the truth of the matter, these patients in the 80s in Italy are not really representative of the United States population in 2022. A lot has changed. The disease etiology distribution has changed. We have the obesity epidemic. We now have treatment for hepatitis C. Alcohol use disorder is more and more increasing. So really, there's a lot of effort that we are putting into using newer data to identify how these states change and what is really predictive. And this is science out of the epidemiology realm, and the newer data that are available also with newer, more sophisticated methodologies will probably let us outline better natural history of cirrhosis, but also identify people at higher risk as they're presenting with certain complications. We have started investigating and working with Neil Kelleher to do where I think the science will ultimately go. What I've been describing is really epidemiology. It is risk prediction using available data, but that's pretty late in the game. And I think where there's a lot of opportunity for us is collaborating with across scientific domains, such as translational research, basic science research, and combining them with the clinical science we have created and are creating to create signatures for decompensation, signatures for mortality, early signatures such that we can intervene, because there are ways to intervene, and signatures for HCC development. Those are, again, tumors that can be treated if diagnosed early on. And I think having precise phenotyping and opportunities to identify risks with blood draws will be very enticing for the future. Thank you.
Thank you very much, Dr. Ladner. Very exciting work and clearly opportunities for advancing the field. Next, I would like to introduce our moderator for this first session, Dr. Susan Quagan, who's chief of our Division of Nephrology and Hypertension in the Department of Medicine. She's also the director of the Feinberg Cardiovascular and Renal Research Institute. And Dr. Quaggan will lead our conversation now for the next several minutes. Sue.
All right, thank you very much, Doug. And I want to thank all of the speakers for really phenomenal presentations. So I'm going to ask the attendees, if you have a question, you can ask it in the chat. You can also raise your hand. You can unmute yourself and ask the question. So I would encourage everyone to ask all those burning questions. And I'm also going to ask Dr. Nadig to turn on his video camera again, and Dr. George. And we already have some questions that have come through during your fabulous presentations. And one that really, I think it could be addressed by everybody. And the question to each of you, and anyone can jump in, including Dr. Vaughn, what is the biggest opportunity that you see in your own areas of expertise for collaboration between CLP and Feinberg? So I'm not sure who would like to start off. Maybe I'll ask Dr. Vaughn there.
I think there are a lot of areas for collaboration that are very exciting right now, Sue. I think, for example, in the Longevity Institute, collaborative opportunities around using multi-omics approaches to identify and clarify precisely what biological age looks like, and that might be a combination of top-down proteomics, epigenetics, metabolomics, and even wearable devices that could guide us as to defining what someone's biological age is as opposed to their chronological age. I think this is an enormous opportunity for Northwestern across multiple different domains, and I hope this is one area where we can engage that kind of talent and creativity to impact upon the field of aging and longevity.
And Doug and I have talked about this before, Sue, but what happens to one's age if they have failing organs? Do they biologically age? And then when you replace that with young organs, can you reverse time? And it's kind of interesting, we all anecdotally, and you're a nephrologist, anecdotally, you see that people look older than stated age when they have end-stage renal failure, but then they get a young kidney and all of a sudden the color comes back, and we kind of all know that they feel and look younger, but can we actually document that, and can we reverse time with younger organs?
Yeah, I think reminds me of something one of our transplant fellows had mentioned, that a patient swore that his white hair reverted to black without any dye after getting a kidney transplant. So I think, I think there's great potential there. And certainly there's some faculty here, who are interested in that very question. And actually,
we're going to need to bring that patient into our human longevity laboratory and see where he fits on the curve. Maybe there's some samples that were saved before he was transplanted, and we can do some of our analyses.
Yeah. Maybe I'll move on to Dr. Ladner and ask, you presented already some really exciting collaborations that are going on. And of course, and the kidney side, we're jealous that the liver can regenerate. But what additional, and actually one of the questions that came through, just to Doug's point, do you plan to do multi-omics in addition to proteomics? And
absolutely. I think, obviously omics is a buzzword, but for good reason. There is so much opportunity. And I think really what's exciting to me is bringing these different fields together. I think omics is really powerful when paired with good clinical trajectory. So the clinical work that we're doing as clinicians, but then also as we're following patients in large cohorts, and we're kind of having an understanding of their natural path, really allows to have very clean kind of phenotypes to then figure out, are there any omics approaches to find differences earlier in the process? Because if we just do clinical trajectories, we're always a little bit behind, right? And by the time patients present sick, a lot has happened. That's also with cancer. By the time we diagnose a cancer, a lot has happened. As a clinician, I would like to get to actually before it happens, if that's even possible, or really, really early when it happens. And I think that's only possible if you take an omics approach.
Yeah, and from a practical standpoint for clinicians who want to collaborate, with CLP, how difficult has it been for you to initiate this clinical, study? It's additional work. It's, are patients excited by the opportunity to participate?
So the collaborations have been wonderfully easy. Dr. Keller has made it very, very simple, which is wonderful. And I think that's just one of the examples, right? I think at Northwestern, we're at a place where people are really willing to collaborate. Now, in terms of the patients, for our particular investigation, but there is many, we actually used discarded samples, which is an opportunity, by the way. Through COVID, it has become possible to collect the samples that are drawn and that would be discarded without necessarily having to enroll a longitudinal cohort like you would in a clinical trial. So it's perfect for preliminary data. And there are national efforts ongoing that actually prospectively are enrolling thousands of patients with this disease, with cirrhosis. And as part of that, we're collecting samples. And what we're hoping is our discarded samples will create preliminary data for these ongoing trials. I have found in transplant in general, patients extremely willing to participate. Almost all of our patients are in some way or form in a study, be it a clinical study to help them get stronger, frailty intervention, be it in tolerance study. So really the gamut from basic to outcome science. I think with the right infrastructure, patients are very, very willing to be part of science.
Okay, Dr. George, I'm going to call on you as well. And you gave a fabulous presentation and clearly have demonstrated the power of whole genome sequencing for identification of mutations, but also for impacting patient care. So what value do you see? I mean, at the beginning, Dr. Kelleher outlined some really fabulous technologies that are available up in CLP. Is that something that?
Well, I think that the one that I keep coming back to is drug discovery and finding ways to target specific genetic factors that may predispose to cardiomyocyte dysfunction, for example. I mean, in addition to some of the models I described, I mean, we have additional cell lines from patients with well-known monogenic causes of cardiac arrhythmia. And we understand their biology, we understand their physiology. We just don't have a good handle on how we might repair them. So small molecule techniques would be one to reach for. But also, I think emerging is the science around RNA therapeutics. And I think I would really love to see sort of an effort to build out an RNA chemistry or RNA therapeutics limb of CLP, maybe jointly with us at Feinberg, because that's really kind of the emerging therapeutic window for genetic diseases. And it's not just the RNA molecule that goes and repairs the damaged gene, but it's also the delivery. And lipid nanoparticles seem to be a popular way of delivering RNA to cells. And so, that chemistry is way beyond my pay grade. I don't understand that at all. So having some resources to develop nanoparticles and lipid nanoparticle delivery systems into cardiomyocytes and eventually into in vivo would be really, I think, a great advance and a great opportunity. So, and then certainly, Neil and I were bantering through the chat about proteomics, and I do think that we are getting closer to the day when we're going to take the whole genomes and set them aside and say, okay, what goes with that transcriptome, proteome, and the like. And so I feel like that's coming soon.
Yeah, terrific. Dr. Kelleher. Neil, I see your hand is up.
Yeah, thanks, Sue. And yeah, so many things of what Al says triggers good thoughts about how to proceed. And, where GWAS ends off in terms of power, the multigenic, polygenic, I think you said, risk score and bridging from genes or where GWAS leaves off, so genetic variation, getting to the protein level, there is a notion that proteins are proximal to phenotype, and they are critical to help bridge that power gap between GWAS and phenotype, so bridging genotype to phenotype. And just one additional, I'd like to get your feedback on that. That transcends cardiology. I think it's human medicine. And but the other thought I had was as a companion diagnostic to unify the approach to targets. You mentioned one in Brugada syndrome and the MAPRE2 gene. That brings forth a very tactical approach, which I've been thinking a lot about, to go after up to, say, 50 different targets in a cohesive way, where if it's RNA therapeutics, great. If it's existing therapeutic, anchored with your great models, to have companion diagnostics that show the drug is on target and that whole program is translatable directly into a clinical trial. So I opened up two threads there. I'll turn it back to you.
Yeah, I think demonstrating target engagement, especially for intracellular proteins, is a very important thing. So I think any kind of new therapeutic program with a small molecule, particularly, or maybe a peptide, you really need to keep target engagement on the horizon. So I like that idea. I think we haven't really thought about a therapeutic endeavor for the MAPRE2 gene, but it's a microtubule component, and certainly there's a lot of pharmacology about microtubules already known. Everything is, of course, horribly toxic, but anyway, I think that's a really clever idea, and using proteomic approaches for target engagement, I think that's really great.
Yeah, and maybe actually on that, I'm thinking actually about a number of studies that I know the others are working on, but Doug, given aging and some of your other fundamental discovery work on Pi1, and do you see a bridge, specifically there, just as I heard Neil talking, that there could be a way to look at target engagement, since you know so much about that pathway, which seems to be fundamental?
Yeah, because we have this very unique resource that we have access to here at Northwestern, and this is the founder population of old order Amish that live in northeastern Indiana that harbor a null mutation in the gene that codes for Pi1. It looks, for all intents and purposes, that the carriers of this gene, the haploinsufficient individuals, are protected from aging in a variety of different domains. We have iPSCs from genetically defined individuals from that kindred, and those cells can certainly be differentiated into different types of cardiovascular cells and tested for their resilience and their ability to withstand stresses that might drive aging or aging-like disease. I mean, that's just one example. There are a couple other large studies going on in the Department of Medicine that are worth mentioning too. Aside from what Rod Passman is doing and the SFRNs that Al mentioned, we have a leadership role in two massive clinical studies sponsored by the Heart, Lung, and Blood Institute. One is around heart failure and preserved ejection fraction. We're the leading center for that program to define new therapeutics for heart failure with preserved EF. That's led by Sanjay Shah and Sadia Khan. And so the point there is to find a way to treat that disorder. It's very, very common. And then Ravi Kalan in pulmonary and critical care leads a Framingham equivalent for chronic obstructive pulmonary disease. And they include deep molecular and clinical phenotyping as part of their program. And there are going to be opportunities to discover new pathways and targets that can either slow the progression slow the progression of chronic obstructive pulmonary disease. So there are a couple other examples.
Yeah, it looks like, Neil, you're going to be really busy.
Great.
I did want to ask, circle back to Satish. I mean, the work is phenomenal and some of the outcomes. So what about, given your mitochondria, like that's pretty dramatic morphologic. Have you thought or have you used it in any kind of drug screen? I mean, it would be a readout for identification of new therapeutics.
Yeah, so we are, this is really recent data. We just published this a couple of months ago in AJT, but what we're looking at is what does it do on other cell types, and what's actually happening when you change mitochondrial morphologic dynamics to antigen presentation. So one thing that was really interesting to me is in parallel, what we found when organs are in preservation phase, the endothelial lining of the vasculature is broken down. And so you actually are exposing subendothelial tissues. So when they then get reperfused, you have these circulating memory T cells that then can go attack these organs more robustly. But what if we actually bolstered that endothelial barrier integrity? Can we make them more immunologically healthy, if you will? So we looked to see what was happening at the level of gap junctions and connexin 43 in particular, and found that connexin 43 gap junction protein is degraded in the preservation phase. But in early preliminary studies, when we were able to bolster connexin 43 and improve barrier integrity, we were able to allow for a longer term survival. And so what happened, all that takes energy. And so when we change the mitochondrial dynamics, what's happening to the level of endothelium? That's one set. So that's kind of what we're pursuing right now. And what are ways we can target that? And to go to Al George's comment before, I just submitted I just submitted an R21 that was being it got a 30 impact score. The pay line, I think, is 28. So let's see what happens. The advisory council met yesterday, but one of the aims is to deliver mRNA-based peptide for connexin 43 as an mRNA therapeutic, not only drug discovery, but drug discovery and targeting, drug discovery and delivery through an mRNA-based peptide that can upregulate these gap junctions to allow for better cell-to-cell communication, because we're all healthier when we communicate better.
Yeah, no, exciting, and congratulations on that score. That's awesome. We have a question, and Dr. Tipu Sadiq, do you want to unmute?
Well, thank you very much for allowing me to ask this question. It's for Dr. Nadig. So I was delighted to hear that transplant patients are happy to help out in research. My question is that when you have donor livers, and maybe this question goes to Dr. Ladner too, and recipient livers, do you genotype them, or do you have availability from their blood samples for genotyping both the donor and the recipient?
So I can take a stab, and then Daniela, we should hear your thoughts too. The answer is twofold: no and yes. So no, we don't, but yes, we have the availability, and actually, it's a very underutilized tissue source of donor procurements. 40% of organs procured for transplantation go unused. That means they go in the bucket. These are great opportunities to utilize these organs for genotyping, for targeted drug delivery trials, et cetera. And so I think we are not doing a good enough job as a field to utilize these types of organs for research purposes because they're there. Right now, when you heard all in the news about the pig kidney going into the human and pig hearts going to human. So the pig kidney model they're using a decedent model. So they take pig kidneys through Regeneron and CRISPR gene editing techniques through the company Regeneron, and the kidneys are going into brain dead donors. And those donors are being those are the model is in the brain dead donors. And so a step before that, which we kind of skipped, is utilizing unused organs for that go unused for transplant.
So I don't know if Dr. Ladner has anything to say, or especially about the liver. Or...
Yeah, I mean, I would answer similarly as Satish, yes and no, right? Or no and yes. We don't create banks, but we do get biopsies and have access to those tissues. And we often have biopsy materials that could easily be used to do all kinds of investigation. It's actually a really rich resource that has been terribly underutilized, in my opinion. And so, I often hear the issue is the tissue. There is no issue with the tissue here. We've got it.
So my personal selfish lab interest is to look at what is produced by the liver versus what's in the brain. As you all know, that the cholesterol metabolism is completely separate. I mean, you can get exodus from the brain, but not input in any significant way in terms of certain proteins, lipoproteins. So that's my interest. And so it's very easy. We have identified certain mutations that would easily lend themselves to looking at the donor recipient genotypes and also looking at the spinal fluid to see if there's invasion of that particular genotype. I don't know if that's possible for us to work together on, and how do we proceed with that if possible?
So everything's possible. If it depends a little bit how easy it is. If you're looking at this metabolism while patients are sick and don't have a transplant yet, then I assume that's kind of where you're going. The tissue.
Actually, post-transplant, we're interested in the donor as well as the recipient material, but also in the spinal fluid post-transplant, because that's when the genotype is different. Only those subjects would be useful for that differential expression identification.
So getting tissue of the donor and recipient is fairly easy of the liver.
So that would identify the subject, because it would be like, a few % of cases that would have a differential in terms of the genotype. Only then you would look for those particular subjects for further studies.
Spinal taps will be hard.
We do them all the time. Never missed.
In an immune, nothing's impossible, and we'd have to talk a little bit more about.
Sure.
In an immune suppressed population, for diagnostic reasons, it would be hard.
Mainly for research purposes. I mean, it's completely for research. There's no benefit to the subject.
Well, I think we've had a fantastic discussion, and I believe we're a little bit over time for the break. So I'm going to thank all of the presenters, and I'm going to hand it over to Dr. Kelleher.
Thanks, Sue, for that moderation. And thank you for all of the clinical insights. That is super important to focus the mind and get updated information. So I know your time is incredibly valuable. That is just, to me, this is just incredibly useful time. So thank you so much for it. We're going to reconvene at 15 after the hour, so 5:15. to flip the script, as it were. And I do want to try to play matchmaker here too. So Xiaoyu Zhang, I want to give him a shout out. He's very interested in T-cell biology, so I would suggest Xiaoyu and Satish could catch up. He's got a great way to try and drug the undruggable proteome, and that's also true of Shana and chemical proteomics. So I'll come back to those themes, trying to connect us up tighter and better, and we'll see you in 17 minutes. Okay, so colleagues, welcome back, and thank you for returning. And I will be offering libations this evening, thanks to our advisory board of CLP, and particularly Stephen. I thank him above all for prompting this and getting us going. It's a great forum. And so let's focus on the technologies that are in development and available. And let me move right into that. And I think as we update ourselves about the unmet clinical needs and new colleagues that have come in to Feinberg and what's happening, let's do the same now for Evanston and what's changed under the CLP umbrella. So the convergence, that term means different things to different people, but certainly we saw evidence of how modern genomics is transforming research and clinical medicine. That as a leaping off point is something it clicked for me when I went to the retreat, the Feinberg retreat on Feb 8, and Rex gave this beautiful overview about Northwestern's medicine operation and what we should be thinking about in the next 5 years. And I came away with genome-informed research. And so I think that's one thing that stuck with me from February. And so having genotyped consented cohorts, all the other aspects of what was said there so well in updated form today, that sticks with me. So then those developing transformative technologies and have hardened platforms that are ready for scaling and scaled interactions of interaction with clinical trials. This is something that's viable. And for those who are earlier stage, who are iPS cells and what we discussed, there were several tactical moves there, discarded organs. These are all extremely tactical if you're in an early stage, requiring material and engagement, preliminary data, as Daniela said. So we have then the challenge to take the same scope of what we have in Northwestern and compete more for new funding to try and advance our position in a competitive landscape, but also really improve patient lives. And certainly drug development is part of that. And so to get to breakthrough, the breakthroughs of the future, we need to bridge genomics with therapeutics for precision medicine. And everyone says precision medicine, but what is the Northwestern, the purple flavor of precision medicine that's local to us, that we can engage and elevate our competitive fitness? So let me give you a sense of that from the proteomics. So now let me focus on Northwestern proteomics. It's about a 60-person operation, and in the last few years, we've moved a national proteomics center to be under Division of Medicine. And I really want to thank Eric and Doug for helping accomplish that and gaining increased space and ability to engage with clinical cohorts. So I've been at this a long time, 20 plus years, and instead now of aspiring to say that the way that Northwestern does proteomics, this top-down flavor, which is to directly measure proteoforms, to have it stated that it is going to be improved. I now have proof in the language of statistics across 5 different disease areas. And that's why I moved to Northwestern. And so in neurodegeneration, cardiovascular health, infectious disease, cancer, and immunobiology, we've been deploying this technology. And I don't have much time, so it is a differentiating approach that we are the international leaders in this area. Proteoform analysis is starting to really pick up steam in across Asia and in Europe. And I'm determined that we maintain our leading position. And so I wanted to pick up on like what Al was saying on when he was talking about genome sequencing and GWAS. Oftentimes there's not enough power to really nail down in multigenic complex phenotypes in outbred populations of patients, what are the targets that we can go after? So part of the vision for the future of CLP is to get alignment and depth with target engagement was one of the things that Al said. So can we use this differentiating technology to actually exploit the fact that proteoforms, that is the complete molecularly specific form of a protein, the entire composition asserted with confidence that that entity, a proteoform, is proximal to phenotype. So we need to exploit that. And let me show it to you. I don't have to promise it. I can show it to you in dozens of different cohorts. This is 2022. This is not 2012. So here's target engagement, Al. And this is in the context of melanoma. So we have wild type MEK1. It's a protein in a pathway that's well known, right? So how are we going to get in and compete for resources to do our science? Well, what you're looking at here in the lower left is the proteoform signature of MEK1 with its phosphorylation. So this is intact MEK1, the whole protein, and occurring in 4 different proteoforms. So different phosphorylated states. And that's the wild type condition. And then if you treat, so someone has melanoma, if you treat systemically with vemurafenib or trametinib, you get a suppression in the phosphorylation. And it's not a Western blot, it's complete molecular specificity for the target, which these drugs target MEK1, which is in some signaling cascade important and upregulated in cancer. So that signature is quantitative, it's reproducible, it's longitudinal in a single patient. So you could really start to aspire to precision medicine longitudinally across a single person or in cohorts. And then when relapse occurs, you have this now hyperphosphorylation. It's coming back with a vengeance. And this alanine 375 mutation is part of the relapse, also with KRAS mutation on board as well. And you see this hyperphosphorylated state. So the signature is done, where you have knowledge of the mutation as well as the allele-specific modification patterns, which can only be asserted through top-down proteomics and proteoform measurement. So we have now a really active last 3 years. We've had some breakthrough technologies. I'll end on this next slide, or at the bottom bullet here. I'll talk about that in the language of technology. But together with Transplant Center and with then years ago, Mike Abakasis, and then particularly Josh Levitsky, you know, he believed in our technology, and we were rewarded with a science paper, but also a U-style grant that is active for the next 7 years. We've mapped over 100,000 proteoforms in 5 different human tissues as part of HubMap project, a flagship Common Fund project. Looked at IgG repertoire with single clone resolution, so I can resolve all the different clones of IgGs, and that was stimulated by the pandemic in deep collaboration with people in the Transplant Center, as well as John Wilkins and Prev Medicine. I've got associations in several apolipoproteins together with Don Lloyd-Jones and also John Wilkins associated with cardiometabolic health. So I could show the same sort of thing in populations of people with varying cardiology risk for cardiological event. And then this technology is we now can image directly proteoforms at 40 micron resolution. And this has been enabled by single molecule mass spectrometry that is called direct mass technology, which we co-invented and co-launched with Thermo Fisher Scientific in June. And so now we can image proteoform specific imaging, and we're pushing this down to the single cell level. So that's all leading up to say that Northwestern can lead the nation in an initiative to sequence the human proteome, that is to determine the proteoforms as they occur at a given depth, and I'm happy to discuss this at length, but we're not afraid to be great and saying to the world, look, we need the analog of the Genome Project. So I'm saying to sequence the human proteome, that is to determine proteoforms and create the definitive reference proteome like we did for the reference genome some 20 years ago. So before we unlock that minimum $1.3 billion 10-year effort, what I'd like to do is partner and scale that partnership with Feinberg in the language of specific targets and of strategic interest. So that could be Pi1 in aging. It could be the targets of interest to Al. And we can get the same data types, and we're getting increasingly facile at doing this across large precision phenotype cohorts. And that's part of what I wanted to say today, focused on proteomics. But we also have many who are doing chemical proteomics at the Institute. So you'll hear from Shana. I mentioned Xiaoyu Zhang. They have incredible platforms to also apply in a similar way to precision phenotyped and genotyped in a targeted way, or to use the power of discovery science also. So, and with Rick and Regan and Carl, of course, drug development is a big part of the history of the Institute and will continue to be robustly into the future. So with that, let me connect over to the next speaker in the session, and that's Vadim. Now, I heard that Vadim might be running late. Whoa, there he is. Beautiful. Vadim, please take us into the world of imaging.
Thank you. All right. It's a really great event, by the way, Neil. So thank you so much for putting together, much needed. And it sounds like we are celebrating the convergence between CLP, physical sciences, engineering, and the world of medicine. It's really fascinating. And I think there are so many opportunities here. The future of medicine, I believe, is at the convergence with physical sciences and engineering and chemistry. So a few years ago, my slides disappeared. Should I share my screen?
Yes. Are you? We have redundant slides as backup if you need it.
Okay. Okay, because sorry, I thought you were showing the slides. No problem. Okay.
Good. Real-time adjustment, Vadim. Thank you so much.
Good. All right. Okay. So a few years ago, let's see, just 1 second. Right. No, that is correct. Okay. So, all right. So a few years ago, we started the Center for Physical Genomics and Chromatin Engineering. And since that time, the center has acquired a secured NIH center grant. So it's now NIH funded, NCI funded, U54 center, and the training program, so the first training program in physical genomics in the country. So we are very much excited about this. And what the center collective is trying to accomplish is to figure out how to reprogram the function of cell genome, not by changing necessarily the genes themselves, which is what CRISPR does, but by restructuring the organization of the genome, of the DNA inside the nucleus. So all of us are familiar with the genetic code, which is the first level of how information gets transferred in our cells. In the past few decades, the interest also has shifted from just understanding genes and their mutations to the epigenetic regulations, things such as histones that regulate what kind of genes need to get expressed or not. And very recently, in the past decade, literally, there was an explosion of our understanding of how the conformation or the structure of the genome might regulate, again, which genes get expressed, which genes don't get expressed. Again, just having a gene does not do the job. You have to express it, and that's what the epigenetic regulation does. I'm sorry, I like to use a lot of analogies. Well, here is one. Let's say you take DNA from a single cell, you magnify it 1 million times. It will turn into 2 millimeter in diameter noodle, and this noodle will spread all the way from Chicago to Dallas. Now you take that noodle and you put it inside an office, like Silverman Hall office. And that's how the nucleus looks like, that's how a genome looks like. It's a very complex, fairly dense structure. And depending on how this noodle is folded, turns out global genomic processes can be changed, and therefore cell phenotype and cell behavior can be affected as well. So here's another analogy. Genetic code is a piece of hardware. Then kind of conventional epigenetics represents programs, apps that run on the cell phone, and the genome structure is the operating system. It doesn't necessarily tell which software to run, but it creates conditions where all software gets operated. So the only way to really understand this system is through convergence of physical and biomedical sciences. When we talk about genome structure, and word structure is critical here, we have to be able to measure it, we have to be able to image it. And that's why one of the fundamental technologies in this field is nanoscale imaging and molecular imaging. The second technological pillar is molecular modeling, being able to model gene expression through the first principles of physics. And the third pillar consists of several transcriptomics and computational genomic analysis. We take this fundamental platform, technology platform, and we deploy it to understand questions address questions in physical molecular biology. And finally, this gets translated for disease diagnosis and therapeutics. Some of the most critical questions that we in the center are trying to address are such as, what is the structure of the genome? How can it be regulated? And how do the changes and how does the change in the genome structure can translate into differential transcriptional systems? And then these fundamental questions are being deployed for translational projects such as cancer diagnostics, cancer therapeutics, and regenerative medicine. Just to give you some of the ideas of the imaging that we have been developing, in this case in collaboration with Vinayak Dravid, who might be here in the audience, is here's an example of chromatin scanning transmission electron-microscopy, the first technique that allows us to see where how chromatin actually looks like. You can actually see individual DNA strands, how they're wrapped around in three-dimensional space. Nobody has ever seen this before, and now we have the ground truth. equally importantly, this information gets used to model transcriptional reactions using first principles of physics. And here, the critical pioneer of this computational technologies is Igal Slifer, a good friend and a colleague too. So we are beginning to understand how this noodle in the room looks like. Even though it may look random, it's not. Genome is organized in a variety of what is called domains that regulate transcription and get regulated by transcription. One of the critical aspects that this structure regulates is transcriptional plasticity, or how easy it is to move the genome from a homeostatic, from one state to the next, for example, in response to a stress. And this is, of course, critical for almost anything that is related to cancer. Cancer cells are masters of adaptation. That's why it's so difficult to kill them with almost any kind of therapeutics, because they change. They are able to change so much. And changing genes themselves is expensive and time-consuming. Accumulating the right mutations is difficult. So what cancer cells do first, they begin to adapt at the epigenetic level, are able to survive long enough to begin to accumulate mutations. So we can deploy this for cancer diagnostics by finding this abnormal plastic genome structure inside cells. And these are the cells that are more likely to be cancers. We're currently, this technology is in clinical trials right now here at Northwestern for lung cancer diagnostics by a very simple swab of the cheek. There are other diagnostic applications as well. Now, in addition to just doing disease diagnosis, can we actually restructure the genome and therefore reprogram cells, make them, for example, less or more susceptible to transcriptional reprogramming? And this can be accomplished by three ways: pharmacological, which is the center of, which is kind of the core of our effort, drug development, mechanogenomic, and finally electromagnetic. So just to give you one example, we can now have peptides or small molecules that can go inside the nucleus and reorganize the genome structure. That drug by itself is not going to kill a cancer cell, but it's going to prevent it from being highly plastic and therefore mutable. And so when these agents are combined with conventional chemotherapy, there is a over 90% increase in efficacy in animal studies done here at Northwestern in collaboration with the developmental therapeutics core, with a very significant reduction in tumor burden. So the combination therapy, you don't allow cancer cells to adapt to other therapeutics. An opposite problem might happen when you want cells to adapt a little better to things such as ischemia. If you are looking at heart cells or neurons, you don't want them to die. You want them to be more adaptable. We are working on these problems as well. Another similar problem is a project that we're doing in collaboration with CARE, Center for Advanced Regenerative Engineering, led by Guillermo Amir, on stem cell reprogramming. By administering certain drugs, or mechanogenomic approaches, we can make stem cells differentiate much faster and in a more directional way, leading to, for example, tissue regeneration, such as bone regeneration. Another example may involve reprogramming whole organisms. We have an interesting collaboration with Professor Marcelina at the Civil Environmental Engineering, where the goal is to epigenetically reprogram coral reefs, coral cells, in order to make them more resilient to heat stress that is literally killing the corals as we speak, with a tremendous negative consequences for the marine environment. So just to conclude, I think we have an opportunity here to converge biology, data sciences, physics, and engineering, again, in the context of CLP-Feinberg collaboration for cell reprogramming, genome reprogramming, for disease diagnosis and therapeutics, including cancer, regenerative medicine, and more generally, to increase longevity. Thank you.
Great. Yeah, thanks, Vadim, and thanks for the adjustment there on the fly. We're going to take Q&A at the end, just before we break and gather for dinner. So we'll turn it right over to Josh Leonard and engineering approaches to the investigation, design, and construction of biological systems. Please, Josh.
Great. Thanks so much. Is the share working now? Looks like it's not quite.
It is.
Well, I think we see.
You can see it?
It looks good, yeah.
Okay, great. I think that's just a Zoom thing. Well, thanks so much. That was super. First, Vadim, I really enjoyed your introduction there and the tricorder reference at the end that you didn't point out explicitly, but that was super. It's a really great analogy also to, I think, some of the things that we do a lot in the story I want to tell you about today from our team. So I thought, given the point of this workshop, I'm just going to give a couple of stories that are interconnected and I think are mainly designed to introduce myself and my lab, the kind of problems that we like to work on, some of the technologies that we've developed. That's the analogy I was mentioning before. And I think more importantly, how those might be used to solve problems that we don't know are actually important problems, but you might know are important problems. And so I'm trying to elicit some of those questions through the couple of narratives that I'll describe here today. But by way of introduction, I'm a chemical engineer by training. I did my postdoctoral fellowship in immunology, and my lab now leads a field of mammalian leads projects in the area of mammalian synthetic biology, where our goal is really to try to just accelerate cell therapies and gene therapies and the developments thereof writ large. So I'll tell you a few stories about what we're doing in those spaces and some of the key applications that motivate our work. I'll start with a disclosure that some of the work on the last vignette I'll mention to you today involves a technology suite, which we have recently commercialized into a company called Cyanix. And this will actually reference back to some of the stuff that Al mentioned at the earlier portion of the meeting today, looking at delivery of RNA and other things as an opportunity. Very much the lab and the company now is focused on trying to do things like that. And there are a lot of opportunities for working together. So like I said, I think if you imagine the field of cell-based therapies, one of the things that people mostly know about this space is that it takes a really long time to go from a great scientific insight to a candidate therapeutic that can be tested in a relevant animal model, scaled up for manufacturing clinical products here. And so in a short version, I think the way that the field of synthetic biology can help, and mammalian synthetic biology in particular, is to catalyze and accelerate that gap between ideation and coming up with something, to spend less time in the engineering itself and to spend more time testing the ideas and seeing which ones are good and moving on from the ones that are not. And so I'll talk to you today about how this field has matured from very much a sort of inspired tinkering to something that approaches a real engineering discipline. And that really underpins why we're at a really exciting time to jump into projects like this, where we're trying to actually leverage true engineering approaches to do things that previously were not possible to do from an engineering standpoint. The motivating application for most of our work is cancer immunotherapy. And specifically, we got interested in working in this space around the beginning of the CAR T-cell revolution. And so a lot of the work that we focused our efforts on are, I would say, CAR adjacent. So we're trying to take on those clinical applications where a CAR approach might not yet be well suited. For example, where there is not a unique antigen or not a known antigen that one can use to engineer a CAR, but you'd still like to have a cell-based therapy that activates specifically at the tumor site. And so that design statement has motivated a lot of the technologies that I'll talk to you about today. When, for example, you know that there are markers of the tumor microenvironment that are distinct, maybe in some ways, but not always, from healthy tissues. And we know this from immunology and from clinical observations. We'd like to be able to map that knowledge into a design statement that actually makes the cell deliver, say, immune potentiating factors only when the combination of environmental factors that are specified is present at the site where that cell is. So in order to take on a complex statement like that, you need to break it down into pieces. And that ultimately guides what genes you introduce into the engineered cell. And many of the things that we try to do in immunotherapy fit pretty nicely into this sense and respond paradigm, where we know environmental factors that are present or absent. We know interventions that might be known to work. So for example, if you have a solid tumor, a big tumor, it may be known that there are immune potentiating factors that would be sufficient to marshal the native immune response to clear that tumor. But when it comes to metastases, you can't actually implement that therapeutic modality because there is not a way to deliver specifically those factors to metastases yet using existing drug delivery technologies. But we can engineer cells to go all places, including metastases and healthy tissues, but then sense and respond to those locales and deliver the mediators only when they're in the tumor sites. So we spent a number of years thinking about what are the required components to do sensing, processing, and actuation in order to implement those therapeutics. One of the areas that I'll start, one of the high-level slides I'll talk about here is that we've gone a long way in this field from thinking that biology is too complicated or not understanding why designs don't work to having a pretty good sense of what that challenge space looks like. And I'll just kind of highlight that with 3 terms here that I think highlight why we focused on the things that we have. The first is that despite what most engineers may think, you actually need a lot of fundamental biology research in order to do a lot of this work, in the sense that every time we try to build a technology, we end up learning something new about the biology as well. And that's part of this virtuous cycle, really, of designing to build and building to learn as well. Burden is a way of summarizing the practical limitations on what you can do. So many ways you might imagine complexity is bad, right? When you're trying to make something practical, reproducible, manufacturable. Another way you can imagine that as a constraint is that there's only so many things you can do to a cell before you start losing some of the natural features that are the reason you pick that cell as a chassis in the first place. And we've gotten much better understanding what those barriers look like and how that can shape the designs that we try to build in these systems. And finally, the variation is one of those things that's a universal feature of all living systems, and probably anything about that size scale is going to have some intrinsic variation. And one of the things that's evolved in some of the work I'll show you is thinking about variation not as something you need to ignore, hope it doesn't mess up your designs, but rather just engage it from the beginning and build designs that work with variation rather than despite variation. So just as a mention of why we think about this all the time, there is no cell-based therapy that will be able to, or that will be made without variation in mind. And just so you have a kind of sense of which variation we're typically thinking about, there's donor-to-donor variation, there's recipient-to-recipient variation, there's variation within each batch of cells, and there's variation in the environments that those cells will experience when they are in a recipient. And so we actually need to think about all those sources of variation when designing an intervention that's practical and that has the ability to perform as desired when implemented. So at the first level here, we as a field have made a lot of progress at coming up with sensors or receptors, natural receptors derived from natural receptors that can carry out that sensing and signaling operation in a way that is applicable to the application of interest. And oftentimes the first pass is to use a natural receptor. And I think what we've learned in a lot of applications is that crosstalk and a lot of other things that are present in natural signaling pathways often preclude the use of natural receptors for doing cell engineering applications. The CAR T cell field is probably one of the great ways of looking at sort of the benefits and costs of implementing native or non-native features in a receptor signaling system here. And the point of this slide is that this overall field went from having no synthetic receptors to having a bunch of synthetic receptors, which is where we are today, each of which has their own unique advantages and disadvantages for different applications, something that we've worked a lot on here. One thing I was just going to highlight for today is that we actually know that even though there are a lot of different systems now, they're all subject to similar challenges. One of the biggest one of which is that they can be kind of finicky. If you express them at a high level or a low level, they don't work as well as you would like. And this in some ways differs from the way the natural receptors signal. And so this has been something that we've worked on for a while and made some progress on. So I'm just going to show you one story to kind of highlight what that looks like. We built a design. We came up with a design for a synthetic receptor, which briefly I'll explain how it works. This is a 2-chain receptor system that when those 2 chains bind to their ligand, it causes a split protein to recombine on the cytosolic face of this receptor, generating a protease that self-cleaves and releases a transcription factor to act in the nucleus. And so this is a self-contained receptor signal transduction system. And we have built things like this in the past. This one was predicted to work okay. But the first thing we found when trying this out was that the split protein component of this just didn't work the way we hoped it would. And generally, you want these things to come together when they are dimerized and not when they are not dimerized. And a general challenge in these split protein systems is that when that doesn't work the way you want, you're kind of stuck. You either have to go back to the drawing board, do a giant screen, something that's pretty laborious here. And so we invented a general approach for solving that problem, which I won't go into detail here today, but it's a computational protein design-based method for tuning split protein reconstitution. And since many of us use these in different ways, I'm throwing it out as a offer that if you have a system that doesn't work the way you want, we've got pretty good tools for making this work now in a way that you'd like. It's based upon integrating computational design with some experimental screening here. And just to kind of in short notice summarize here, the original version of our designs signaled as shown by this black and gray bar, which is to say it didn't work at all. The off and the on states are identical. But with this split protein optimization system that we invented, we can generate the most high-performing synthetic receptors ever reported in literature to date. So I think the point here is that by using these engineered systems, they may start off being worse, and oftentimes are, than natural systems. But because the mechanism is interpretable, you can use design tools to optimize performance in a way that makes these things really attractive for both fundamental research and for translational applications here. We've also done some work to show that we can solve a related problem, which is that depending on how you plan to deliver your gene to your cells of interest, whether it's a lentivirus or a transposon or something by electroporation, those all impact the way these things work. And the nice lesson is if you design things to be inherently robust to expression, they just work. When you put them in new cell types, when you deliver them by different methods, it becomes a lot more generalizable if you apply this principle. So this is one of several stories, but I think a really good lesson to think about where that tech is driving some utility here. I'll end with just 2 quick stories, one of which is that we've applied similar approaches to the next layer here, which is in information processing. We built a library of synthetic transcription factors here, and you did the normal thing to kind of characterize how these work. If you simply do those experiments, you end up with a library of parts that are useful for kind of picking the big one or the small one or the strong one or the weak one. But one of the big things I'm proud of our field in general for having done now is moving from this kind of descriptive approach to one where we use computational models that are pretty sophisticated to do not only description of how these things work retrospectively, but predictive design. And I think I'll skip over this other than to say, we can now design circuits that work the first time for many different types of applications without any tuning or iterative refinement necessary, and at levels of complexity that are far in excess of what we actually want to try to build from a translational standpoint. And the last 2 slides I'll show is that we're really close to actually getting to this idea of CAD-based work in cell therapies. What we need are great ideas from clinicians, from folks who have questions. These can be basic research questions as well as translational objects. We have 3 or 4 translational uses that I'm not showing you today where we've taken these approaches and are employing them to build cell-based therapies and evaluate how they work. And I think the last thing I'll show in the interest of time is one slightly different story where we are using synthetic biology to engineer cells still, but in this case, not to make a cell-based therapy, but rather to make a cell-derived product that can serve as a delivery vehicle. And in fact, this is where our commercialization has come in. These vesicles are called exosomes or extracellular vesicles more generally. There are a half dozen labs, at least at Northwestern, who work scientifically in this space overall. And what we've done over the last 7 years or so is build a suite of technologies so that you can start thinking about these biological particles in the same way that you would synthetic particles. We can load them, we can direct targeting, we can do delivery. And we recently showed, for example, that you can use that to deliver Cas9 to primary human T cells. And we've done extensions of this to mRNA, gene delivery vectors, and so on. So I think I'll stop there, give over the time, Neil, and we'll take Q&A at the end. But thanks very much, and I look forward to chatting.
Josh, thank you. Yeah, really some resonance points there on delivery. And I think about trying to align technologies on targets and systems that that are of highest interest to Feinberg, and that synergy could be accomplished, but only if we communicate. So please, now we'll move it over to Shana Kelly and a new high throughput platform for cell profiling. Please, Shana, take it away.
Thank you. Thanks, Neil. Really appreciate you putting this together. And it's just been phenomenal to listen to everybody and get to know, put some faces with names. I came to Northwestern about a year ago. I feel like I've just scratched the surface of kind of getting to know the community here. And I'm really delighted to be part of all the great things that are happening at this institution. So I'll just tell you quickly today about one of the things that we do. We really, in my group, develop new bioanalytical platforms. We do a lot of sensing, and we're increasingly moving into in vivo sensors and being able to look at biomolecules in real time with embedded sensors. We also have some work. It was great to hear a little bit about mitochondria and transplantation medicine. We have had a longstanding interest in mitochondria and have developed some delivery vectors for molecules that we can use to modulate mitochondria, to learn more about how they work. But today I'm going to focus on cell profiling and a platform that we've developed there. And the idea behind this was to come up with a way to look at very large collections of cells, find rare ones that have interesting phenotypes, maybe a disease phenotype that we've modulated somehow, and to really push past the limitations of things like FACS, where you have incredible precision in terms of isolating cells, but it's slow. It's hard to deal with 1 billion cells. It's hard to deal with biological samples, blood, associated tumors, real samples coming out of patients. And so we developed this approach called magnetic ranking cytometry, where instead of using fluorescent labels, we use magnetic labels. And I won't go into the details, but we can basically count magnetic particles the way that a flow cytometer counts fluorescent antibodies. And through counting the particles, we can make very precise measurements of protein expression. And we can do that and use that information analytically if we have an interesting problem that we want to look at. The data that comes off of this platform is very similar to what you get off of flow cytometry, but you can look at 10 cells in a background of 1 billion. We've embedded this approach in clinical studies. We've looked at rare immune cells that we can use to screen patients for mesothelioma. We found other kinds of rare cells in the blood. And so this is something that we deployed widely as an analytical capability. But it's turned out that what's even more useful about this is using it preparatively and using it like you use a fax machine, but with the ability to process 1 billion cells in an hour. And so we do that. We take labeled cells, we sort them into devices where we just bin them into compartments according to protein expression or other phenotypes that an antibody can help you read out. And then we can walk away with hundreds of millions of cells that are a particular subpopulation. So that's the approach and the technology. And again, it is a souped up cell sorting type of technology, but it turns out there's a lot of really important problems that one can deploy this on. And I won't talk today about our cell therapy work. We were able to use this to identify really a new cell type in the blood that has tumor reactivity and can be used to treat solid tumors right now in mice, but we're hoping to go into humans eventually, and just launched a new company. I should have had a disclosure slide like Josh did, but the name of the company is Controlled Therapeutics, and that's getting launched locally here. But what I want to spend my remaining 6 minutes on, or whatever I have left, is using this as a tool to really drive high throughput, high efficiency identification of new therapeutic targets. And this approach, we call this leapfrog. So we're doing large scale phenotypic functional genomics at scale here. We're also trying to just leapfrog over what's currently available. And so basically what we're doing is using the cell sorting approach to sort through billions of CRISPR edited cells, where you have one knockout per cell or one activation per cell, or all the things you can do with CRISPR libraries. And then we target a disease phenotype. It could be the overexpression of a protein. It could be the secretion of a protein. We have a lot of different phenotypes that we've been able to target. And we can take billions of CRISPR edited cells, sort through them in an hour, And this really allows us to scale what you can do with this kind of screen. So we can look at panels of cell lines with different mutations. We can look at families of targets. We can do multiple time points, which improves the quality of the data significantly. And we've been running lots of interesting screens. And this is a platform where we're looking for new interesting disease biology. So if you have a favorite disease phenotype, and you'd like to do one of these screens and you want to do something big and ambitious, we would love to hear from you and talk about how to target it with this platform. We've been quite oncology focused to get going, but we do have some ideas in other disease areas. And again, my group has worked with clinicians really the whole time I've been running a lab, and we love to collaborate. And I'll just run you through a couple of these kind of proof of concept studies that we've done so far. One of the first things we looked at was targeting an immune checkpoint, so CD47. This is a protein that the immune system looks for, and if they find it, it's a don't eat me signal. We did a screen, we found a really interesting novel hit that turned out to be an enzyme that cyclizes a residue that's at the N-terminus of CD47. It turns out that's right where macrophages look to figure out whether the cells have CD47 or not. And so we were quickly able to determine this was a pretty interesting target to take active CD47 off the surface of cells. We found a small molecule. We showed that this was a druggable target. And now we're off to the races in a partnership with Evotech, where we've identified already some interesting novel chemical matter, and then are going to look for a channel to take this forward as a solid tumor treatment. So that was one thing that just got us going. We've recently been doing a lot of work on KRAS. An interesting target, lots of reasons to be able to combat mutated forms of KRAS, take it down in solid tumors. Lots of activity in the clinic with the new Amgen agent that has some success with doing that with the G12C mutant of KRAS, but there's many other mutants to target. And so this is where the power of our platform comes in, because you can do these large scale screens looking at cells with wild type KRAS, looking at cells with mutated KRAS, and you can do those screens, you can do the bioinformatics, and then pick out selective regulators of mutant KRAS, something that doesn't affect wild type levels, but selectively gets at the mutant. So we have this really interesting hit enzyme, or I'm referring to it here as SMKM because we're still getting the IP worked out. But this is now a validated target, something that we're taking forward. We've proved this out in cell lines. It's a protein that's involved in lipid elongation, and we've shown that it's druggable. We have a tool compound. We've gone into animals. So this is another one that we're kind of off to the races with an interesting target. And then I'll just finish with just a few words about another capability that we've developed, and that's to look at regulators of cytokine secretion. And so we asked whether we could take this platform, instead of looking at protein expression or modification, can we watch cytokines being secreted out of cells and read out the efficiency of that process, and then look at that in CRISPR edited cells and try to find modulators. And so we first did this in the context of IBD. And we went through this workflow, did the screen, we catch the cytokines on the surface of the cell, and that's how we use our magnetic particles to read out levels of secretion. Found some really interesting hits. We did a kinome screen here, and so everything that we pulled up was druggable. We went into a model of IBD, drugged mice with an inhibitor of the kinase that was one of the top hits of the screen, and showed that we could rescue the intestinal stem cells that usually get really hammered by the cytokine secretion in IBD. So that's just another example of one of the things we've been able to do. Lots of other stuff going on I don't have time to talk about today, but again, would love to work with people. We think this is really a way to just turn up all kinds of new, interesting targets and to focus on regulators. The tip of the iceberg is, inhibiting small molecules and active sites, but there's so many other things that we could look at as therapeutic targets. So I thank my group, and thanks again for including me today. And I'm happy to take any questions during the Q&A.
All right, Shane, thank you so much. And great examples of chemical proteomics in many different contexts, maybe give some color and context to what we're brewing there at CLP. So let me turn it now over to Carl, and he can moderate the Q&A, please.
Yeah, definitely. Thanks, Neil. Moderating between everybody and dinner. Great place to be. But I want to thank Vadim, Josh, and Shana for some spectacular talks. Obviously whetting appetites for a lot of people. What's interesting is there's been some chatting going on. Sue asked a few questions, but I think we've got one outstanding for Josh at this point. I think Al asked, talking about synthetic transcription factors. And again, he's trying to get it. I mean, there was a lot. You guys did a superb CLP shock and awe. There's so many things that people are thinking about, which is, I think, really one of the primary reasons that, Neil et al. Put this together. But Josh, the ball's in your court. Maybe, do you see the question on the chat?
I
side?
do. Yeah. Totally happy, Carl. Yeah, thanks. So I should have said this. One of the big design principles that we used for these synthetic transcription factors was to render them as independent as possible from native control there. So one of the things we do is try to, in general, we're using these to control native genes. That said, we have been building these kind of biosensors. One story I didn't tell you about was we built a circuit, which is a hypoxia biosensor circuit. And this is a great example of how the native hypoxia response is useful. It can use some of the things you may want to do to, for example, have microenvironmental induced activation of a cell, but it doesn't work exactly the way you'd want. Sensitivity and fold increase for some applications. So we can link together responsiveness to native transcription factors with these kind of gene circuits to overcome some of the limitations of classical reporter design or classical things like this. So that would say we can both take native transcription factors as inputs into these circuits, and certainly expressing native factors as outputs is much more straightforward. I just wanted to highlight that first connection there. But the transcription factors themselves, they're designed to be as small as possible, such that, and actually as weak as possible when bound in isolation, so that they only turn on gene expression when they bind in these little arrays that we build at the DNA level. And that renders them fairly inert with respect to the rest of the genome.
Cool.
So may I have a follow-up? Is your library screenable?
It could be. It isn't yet. So we built this using previously identified zinc finger based proteins, 3 zinc finger motifs, minimal ones there. So you absolutely could use this kind of thing, for example, to find a zinc finger or actually any other protein where you wanted to use that kind of information processing for a screen purpose. We're doing a little bit of that with a different story, not exactly for this application. So I think the answer is yes, but it'd be great to learn a little more about what kind of screen you're thinking of.
Well, just as a background, and this is more of a neurological focused question, but there are a lot of monogenic conditions in which the gene defect is haploinsufficiency, meaning one allele of 2 is gone or just completely dysfunctional. If you had a way of amping up transcription of the intact wild type allele, you might have a treatment. And there are lots of ways to do that. And there's a company on the West Coast called Encoded that's developing, customized transcription factors targeting some of these haploinsufficient genes and neurodevelopmental disorders. But I think, tackling haploinsufficiency is the reason I brought it up. Could it be screened if you had a gene of interest and you could somehow have a readout to inform you that it was being upregulated? That was the question.
Thanks. That's cool. Definitely give it some thought.
Yeah, thanks, Al. Xiao Yu, you had your hand up.
Yep. So I have 2 questions for Josh. Hi, Josh. I'm Xiao Yu. Fantastic talk. So the first question is about the exosome you mentioned at the end. So I was wondering, how you pack those materials in this exosome. Do you pack them in vitro, or you transfect or transduce the producing cells? Later, the cells will generate those exosomes. And can you pack small molecules in exosome? So that's the first question. So the second question is, what do you think of engineered NK cell therapy space? Could those approaches overcome some of the limitations faced by T cell therapy?
That's great. I'll be real quick to spread the time out a bit. So I'll do the second one first. So we have a project. It's a collaboration with Ben Zhang engineered NK cell therapies. So the answer is I certainly hope so. And we do think about those kind of transient interventions as being something that could be a really good complement, let's say, or applicational complement. To the exosomes, as I'll just mention as a for everyone's info as a field, these are about the same size as a virus. And so within that space, some people do purify the vesicles, load a drug with them, and then you can, for example, use the targeting of the vesicle to deliver your drug where you want. That is possible, especially for drugs that can pass the cell membrane pretty well. What we have leveraged more is the biological loading. So this involves some of those technologies I referred to, where we do kind of the same things a virus does, but not with viral parts. Actively loading Cas9 into, in this example, actively loading Cas9 into the vesicle using a new protein loading domain that we invented as a part of that project that drives up the loading of the Cas9, doing so in a way that's reversible so that when it gets the recipient cell and fuses, it can still release and go to the nucleus. So the short version is we work much more with the biologically encoded active loading and delivery steps, but it certainly is compatible with synthetic loading and surface functionalization and things too. We just haven't done that as much.
Great, thank you.
Other questions for this?
Yes, I have my hand up, but it keeps falling down. This is Fred Turek. Can you hear me?
Oh, hey, Fred.
Yeah, thanks. So I'm going to address this question to you, Neil, but it could be addressed to other people as well. I'm addressing it to you because you mentioned personalized medicine. As you know, biological systems have inherent temporal organization. And to use that, if I can use an analogy, you look at me, I'm structurally, you can see my two eyes, my two teeth. You can see my chromatin structure. You can see my proteins being expressed, which ones are being expressed, like the genome. The genome is constant, but the expression of the genome is not constant. It changes on a 24-hour basis. And so I'm just wondering, Neil, with respect to personalized medicine, how do you take into account that there's 24-hour variation? I one time asked that of a leading genomics person who was, going to be looking at the gene expression and disease states in 100,000 people. And I said to him, well, what if you're looking at in the daytime and you're missing because all the action's at night? And he just looked at me and said, I know. So I'm just wondering, how are you guys taking into account temporal organization, particularly circadian organization, in the way you're thinking? Certainly with chronotherapy now, there's a lot of interest in, giving drugs at a certain time of day, which will be more effective than another time of day. And it just, hearing all these talks made me think, hey, where's 24-hour organization in your thought plan?
Yeah, so there's many timescales that flow in biology, right? And there is an aggregate, a detectable steady state on other timescales where dynamics. For example, you can have a dynamic phosphorylation that is extremely, the half-life of which is minutes. But when the drug comes on board and the pharmacokinetics have been worked out, what you saw there with the vemurafenib treatment is an average. And that average is faithfully reporting the overall organismal phenotype. That is to say, a human's response. The drug is on target and it can be quantified down to 5% CVs and relative standard deviations. And despite the fact that there's all these underlying dynamics, if you have a signal that has a through line that can connect to the phenotype that you're after, then you can use good study design principles to go ahead and do your science. And there were many sleep awake cycles for those patients that were on that kinase inhibitor. But nonetheless, you got reliable protein level, that is to say proximal to phenotype. It should be the case that genes and DNA information is remote from phenotype, and that closer is RNA and closer is protein and metabolites. So we're trying to take that next step forward. And so I hope that addresses some of your questions. And we've seen it across in many different areas of human biology, that same, I'll call it steady state. I'm not a kineticist, but it's an average observable that connects tightly, statistically powerfully to a whole human being. Translational proteomics is possible and it is powerful. It just hasn't been done correctly to date.
I would add, Fred, I think this is such an important point. You know, there was a paper that came out in Nature a few months ago where they watched circulating tumor cells in cancer patients, and they found the levels went way up at night when people were sleeping.
I know.
And then they kind of followed it up with all the stuff showing that, like, you know, metastasis is happening more at night than during the day. I don't know that the biological links are there, but I think that is going to come front and center for people like us doing all of the omics to try to take account of that. So it's incredibly important.
Yeah, it means good study design and sampling with high enough density and to just handle that which you seek to study. Sorry, Fred, go ahead.
And I was just going to say that the molecular circadian clock is regulating at least 50% of gene expression, and then I would assume that down the line protein synthesis. At least 50% of the genes in any given tissue are cycling. And the circadian molecular machinery links into the molecular machinery of many, many systems, signaling systems in the cell. And so I just want to put that out there to put so that people who are not thinking about circadian rhythms, which is all I can think about, sleep-wake cycle and circadian, it's the only thing I know anything about. Just thinking that, I hope you will take that into account, at least in your thinking. I'm not sure that we're maybe not at that level for dealing with what some of the systems you guys are working with yet, but it's going to be, I wrote in an article in Science a couple of years ago, there's no such thing as personalized medicine if you don't take into account the circadian sleep-wake variation. It doesn't exist.
Yeah. I guess in some cases it'd be easier to get patient samples while they're sleeping, and some it would be much more difficult. It's gonna have to navigate that.
Yeah.
I would say this is, I think it's a really an exciting idea and a very good question, and highly underexplored. Absolutely, just to understand what kind of genomic changes we have at night. That needs to be investigated. There's so many opportunities here. Absolutely.
Well, we do have a Center for Sleep and Circadian Biology at Northwestern, and I would like to link some of you guys together into this concept. I'm not saying that I could follow what you guys are doing at the it's above my pay grade, as somebody said earlier, but I would hope that we can, Northwestern is kind of known for its research in sleep and circadian rhythms, and like to integrate that with what we've heard throughout the workshop today, and maybe at dinner tonight.
You can sign me in. I'm sold on the idea.
All right.
Cool.
Yeah.
Other questions?
Got about 5 or 10 minutes.
5 or 10. I mean, now's an opportunity for SFM faculty to really, see what some of these platforms that are not only in Evanston, but some really exciting scenarios and platforms.
Just to follow on from Fred, I wonder, some of the circadian master regulators, like to pick up on Shayna's theme, those clock proteins, or they could be targeted and look at how they vary, and then look at the ripple effects after them of the larger, if you wanted to do proteomics or metabolomics, to do a discovery mode. That's how technology developers sometimes think, is in a targeted way. And if you're thinking about grant opportunities, there are ways to address a study design that is not going to break the bank, get preliminary data, show statistical power, reviewers eat that up, and then do something bigger once the funds come in. But that takes a year or two, a building process.
Well, maybe Carl, it's Sue from the, I'd just like to thank all the presenters. It's just phenomenal. And I know the CLP, and we've worked with them. It's been fabulous for, the Kidney Center. And I know, Shayna, you and I am hoping we're going to get something going. But, I still need to process a lot of Josh and Vadim. I haven't heard you give presentations before. I think of a whole bunch of things, but my questions are really naive. So I might reach out to you afterwards. Like Vadim, I'm not sure if you're able with your imaging to identify structural defects. So for example, we have a really interesting mutant mouse that we've been working on for, about a decade. It turns out, the mutation is autosomal dominant, causes cystic kidney disease. It's not any of the known genes. And we've done whole genome, we've done, single cell, all the omics. There's some really interesting findings, yet the point mutation, the only one in the critical region, does not seem to drive. So talking to people, there was a question, is there a structural, it could be, it was an ENU mutant mouse that generated this. So I don't know if you're able at that resolution your images are amazing, but my guess is it's not quite there yet to identify maybe a structural mutation on a single chromosome. Yeah.
Well, so the way to do it would be to identify the locus.
We know that, yeah.
Either CRISPR series or Razorfish. And as soon as this is done, the rest is actually easy, because then you can just look specifically at the chromatin and DNA associated.
So we know it. We know it's in a 300 megabase region of chromosome 6. We know that. But anyway, I'll follow up after. As I say, I've got all sorts of naive questions for everybody.
Well, as the title says, this is an inaugural, get together. I think there's a lot of information and opportunities, and it takes, I can only speak for myself, it takes a while to start seeing how some of my program might interface with others. So I think, again, I want to echo, Sue, what you said, is to thank all the speakers for taking the time to sort of digest down their programs and make it bite-sized, to the sense that to stimulate conversation and discussion. I think that was really the goal of this, is to start discussions. We're not going to finish anything, but to really get people more invigorated in terms of where those bridges can be built or strengthened, right? Or just even explored. Nothing, just brainstorming with people that are, in slightly different camps. I mean, that's one of the great things. It's built into our DNA at Northwestern, right? No pun intended. And I think that we can continue to carry forward with that. And CLP is a real, I think, a meeting place to help stimulate those types of interactions.
Absolutely.
Yeah. Okay. Well, Carl, maybe I'll go ahead and move to finish up then.
Sure.
Okay, great. Yeah. Just remember to get IRBs written in a way where you could take part. We will be trying to line up technologies together to stack them and align them with Feinberg driving biomedical problems, many of which we've heard today. We could strive to do this twice a year, and get back together then in the late winter or early spring. We have a dinner planned tonight at 7:30 at Maggiano's, Little Italy, at the address shown on your screen. I'd like to thank everybody for their time and to stimulate conversation and how we might go after new targets, and that is to say drug and diagnostic targets, but also grant targets, and compete on the national and international stages more effectively, increase our competitive fitness, and do that in a team science way. I'll note that CLP has a lot of grant facilitation, dynamic reserve, so we can specialize in P, U style, large grant activities, multi-PIs. These are things that the Institute has excelled in. And so we're happy to partner to do that. And we've gone after things from $10 to $90 million just in the last year or two. So keep that in mind if you need augmentation or you need to access the folks you saw here or other resident and non-resident CLP faculty that may or may not be within Silverman Hall. And yeah, I think we'll end it there. And please let us buy you a drink and dinner if you are available. Come join us, and just thank you all again for your time and expertise. And see you out on the circuit.